Hello everyone, welcome to the course Python for Biological Data Analysis. Today we are going to cover statistical analysis for biology. My name is Ashfaq Ahmed and I work in the field of structural biology in bioinformatics. So let's start. First of all, you need to open your Chrome or any other browser and just type Google Colab. Google Colab, Colab, enter. You will have this kind of window. Click here. Now click GitHub here and in the search box type ASH100. Press enter. Here, click here and scroll down to Python for Life Sciences. Scroll down again and come to Chapter 8 Statistical Analysis for Biology. Now you need to click here. It will open a new window on your system. Now connect your Colab session to the internet. Connecting. Okay. Learn Python for biological science. This is a kind of heading for this Colab notebook. And this is my information, just a name and about this course. So far, we have covered chapter 1 to 7. If you want to, these are the course content I covered in this course. If you are interested to watch any of them, you can just click here and it will lead you to the YouTube lecture. Okay, so today we are going to cover statistical analysis for biology, which is in the section of biological application and chapter 8. Okay, now what topics we will cover both practically and theoretically today. So we will cover hypothesis testing basics, t-test and chi-square test, correlation analysis, and introduction to SciP states. Okay, so let's start. What actually is hypothesis testing in biological research? So, hypothesis testing is actually a statistical method, and you can also test your hypothesis with the help of statistics. Remember, there are basic two tests to the one we call it null hypothesis, and to the other is the alternate hypothesis. Okay, if a data except or if a data is if a data is okay by null hypothesis or accepted by null hypothesis, then we will use this one. Otherwise, it will be the alternate one. So let's understand what is null hypothesis or H naught. The null hypothesis actually represent a statement that no effect, no difference, or no relationship is observed in your data. Okay, it it doesn't matter what type of your data, what type of your data is. It's from biotechnology or from agriculture science or from botany or whatever. But if the null hypothesis statement is, you have to understand this point that no effect, no difference, or no relationship. This statement is actually representing the null hypothesis. If your data disprove this any of the statement among these three, then you will have or you will use alternate hypothesis, or you can say that my data rejects null hypothesis. Let's discuss further. It is the hypothesis that a researcher attempts to disprove. I hope you will got my point now. Like, for example, you have some data of plant growth. We will say like this. And before running a null hypothesis or before checking a null hypothesis, there is a statement that the plant growth in your data is not affected by the nutrients you used in in the field. We will say like this. For example, you have used some different kinds of nutrients and different kinds of drugs, whatever, or different kind of growth hormones in your data set, and you recorded different growths at different interval of time. So before performing the null hypothesis, the statement will be there that there is no effect of these treatments on the growth. If your data disprove this statement, then it's okay. Otherwise, null hypothesis will be there. It's just like think of it as innocent until proven guilty. Okay. So here I use that example. For example, the fertilize has no effect on plant growth. It's not I'm saying, it's the null hypothesis saying, or the nutrients you used has no effect on the plant growth, or the growth hormones you used does not affect plant growth. If your data disapprove this statement, then it's okay. We will call it H1 or HA. And we will say that my data is disapproving the null hypothesis. But you need to understand in what condition you can say that my data is disproving a particular statement of null hypothesis. Okay, so let's understand the alternate hypothesis first. Um, Designed or designated by H1 or HA, and the statement, it is the statement a researcher wants to prove. Because you perform experiments, you want to prove that yes, this growth hormone has effect on the plant growth. Yes, this fertilizer I used has affected the plant growth. So you want to approve this, but the null hypothesis say it has no effect. So let's see how we proceed further. If your data contradicts H naught, then it will be suggested that there is a relationship, there is a effect or growth, whatever. Okay, the fertilizer does affect plant growth. I'm sorry, I have some throat infection nowadays. The fertilizer does affect plant height, so it will be the alternate hypothesis H1. Okay, but how we will prove this kind of thing, whether my data is rejecting the null hypothesis or not. So remember, we collect the data and then perform a test. We call it t-test or other test, chi-square test, and whatever. Then there is an important term, we call it p-value. And the p-value, you will have some p-value, then you will compare the p-value, the observed p-value, with the significance level. What is the significance level? The significance level is actually 0.05 or 0.05, and we call it, or internationally they call it, the significant level, and that is called 5%. So based on that significance level A or alpha, we make a decision. How? The p-value is actually is the probability of observing some result in your data. And from that probability, we will decide that whether in your data the null hypothesis is true or the alternate hypothesis is true. If the p-value is smaller than the significance level, which I mentioned 0.05, then that means your data rejects H naught. And your statement, the statement a researcher wants to prove, is correct. But if the p-value is larger than the significance level, which is 0.05, then results are not surprising under null hypothesis or H naught, and you are failed to reject H naught, and the H naught statement is correct. The H naught statement was that fertilizer has no effect on the plant growth. The H naught statement was that your data or the growth hormones you used did not affected the plant growth. Okay, and here I already mentioned to you like what is the significance level and what that means, and this is actually the predetermined threshold researchers are using, and this is the significance level, and that means you accept the 5% chances of making type 1 error. I will not go in the details of type 1 error. Okay, so this was the decision rule we discussed earlier. Okay, now we are coming to some basic things. We will import some libraries. If you are following this series, so you will be aware that why we are importing NumPy, why we are importing pandas, why we are importing matplotlib and Seaborn. But today we also add a new import SciPy. From the SciPy package we are importing stats. So we will run all these five imports and let's see. Okay. Now, I always emphasize in this step that in your case, probably you will upload your own data here. You can generate a new code here. Let's say I click here and now you can upload your own data. But in this case, I will simulate, I will generate some arbitrary data and then I will teach you on that data. So I'm using a random seed 42. In our last chapter, we discussed why we use random seed or NumPy random seed because we want reproducibility. Okay. Now I'm simulating or I'm generating two kinds of data sets. To the one I call it untreated and to the other I call it treated. And I'm using npr or numpy random.normal function and I'm using LOC10 scale to size 30. But here LOC13 and scale 2 and sizes are the similar. What does that mean? The LOC or the log 10 means the untreated group has a mean of 10 here, but here I mean the untreated group has a mean of 13. Just like if you think of gene expression data, so this is the treated gene expression data. So the I asked the code to generate some fake or some not fake, yes, fake, you can say, generate some fake data, 30 data or 30 lines of data, and in the treated group, keep the data up on the higher side. Okay. Now, let's visualize this data. First, run this. So when we run this, our data is generated. Now I'm visualizing the data. So what I did in this, I'm using a white grid. This is the grid. Okay. Now, then this is the figure size. You can tweak it like 8 centimeter longer and 5 in height. Okay. Then I'm using the color like to the untreated sample color sky blue to the treated sample the untreated sample color sky blue and to the treated sample color salmon. Okay, for the untreated sample, use this label. For the treated, use this label. I use this option KDE is equal to true. That means that it will add a kind of smooth curve for the distribution. Okay, and then these are this is actually the main title of the chart and the X label and the Y label and then PLT show. So we have this kind of data or this kind of visualization or distribution chart I discussed in my previous chapter. Okay, now we are coming to the statistic, but first I want some statistical summary of my data. So what kind of summary I am interested in? I'm interested in the mean and standard deviation. So I asked the program to print untreated mean and print untreated standard deviation and the same for the treated group. And I used yesterday we also used that NP dot mean, NP dot standard deviation, the untreated, and then the treated data frame and treated data frame. Why I'm using these name treated and untreated? Because if you see here, my data is generated with this variable treated and untreated. In your case, if your variable is different, you need to change that name here. So that says that the untreated mean is 9 and the standard deviation is 1.7 for the untreated and for the treated you can also see. Now coming to perform the t-test. Okay, we are using two types of functions, t-test and p-value. That means we are interested to see the p-value and to perform the t-test. This is the states.t-test and this is actually the function, but for the t-test. But we are interested to perform the t-test and generate p-values both on treated and untreated. Then we ask the code to print the t-statistic value generated by the t-test and the p-value generated by this function. Okay, let's run it. Okay, the t-test says that the t-test statistic is 6.6 so on and the p-value is 1.2 exponent 0.8. So apparently students are making a mistake and they think that okay, this is 1.2, so clearly this is not significant, but this is not the case because I had already mentioned in the start that the significance level or the alpha value is 0.05, this one, and your p-value should be smaller than this value if you want to reject the null hypothesis. Okay, so what do you think? In this case, the p-value is 1.2, so whether it is significant or not. So I have also included a reference sheet for you. If the p-value is larger, excuse me, if the p-value is bigger or larger, then we will call it not statistically significant. If the p-value is smaller or equal than statistically significant. If the p-value is smaller or equal than 0.01, it will be strongly significant. If it is in this range, very strongly, and if it is in this range, this is extremely significant, then you can use these terms in your research papers. You cannot call extremely significant if your p-value is here. That will be called kind of strongly significant. Okay, our p-value is 1.23, but the exponent is 0.8 08. So let's when you 1.23 into 10 raised to power minus 8, then this will be your value or the exact p-value. So now you can think it is extremely significant. So in this case, if you think, if you remember in the start, I told you that the statement of the null hypothesis is that no effect is there in your data. So now our data proves uh, extremely significant from the p-value, then it means our data is able, or our data rejected the null hypothesis. So now we reject the null hypothesis. That means there is a relationship. That means fertilizer played a role. That means the growth hormones played a role. Whatever your data says, you can explain that way. Now, coming to another test, possibly you heard this name, chi-square test or x-square, or some people also call it chi-square test, but that is up to you. We will not go deep in the pronunciation. So this is another statistical hypothesis, and here we examine the relationship between two categorical variables. Okay, for example, we have, let's talk about some genetics now. For example, we have some observed frequencies of mutations. For example, we have two genes, and in one gene we observed some mutation, some frequency of mutation. And what we were expecting before the sequencing, we were expecting the frequencies to be just like how to say, just like the reference genome, but we observed some more frequency, some more mutations. Okay, so the goal of the Thai test is to determine between the observed and the expected result, and to show that whether it could be real or it just by chance. Okay, so types of chi-square test, like I mentioned here, that we checked categorical variables and its association. For example, hair color and eye color. Is there any association? So if you have data, you can run chi-square test on it. Okay. Now, let's understand some difference between the chi-square test and the t-test we observed above. If your variable type is categorical, just like gender, species, or presence or absence, then you can use chi-square test. But if your variables are continuous or quantitative, for example, height, weight, growth, you are measuring these kind of things, then the t-test will be used. Okay, to assess the relationship or compare observed versus expected frequencies, and to compare the mean of the two groups. So if you think a little earlier, like we also calculated the mean of the treated and the mean of the untreated and treated, then again the t-test basically first calculates the mean value, and then generate some, how to say, some interpretation or some results. So the t-test actually compare the mean of the two groups. And actually the chi-square test assesses the relationship or the comparison between observed and expected. Okay. Okay, now what are the application of chi-square test in biological data? You can use it in genetics, ecology, and medical research, and so many other things. But I just coined these three things. You can read it. Okay. Now here to perform chi-square test again, I'm going to simulate some data, and I'm simulating two types of data. One is observed and another one is expected. And this is the data I want to, it's a genotype frequencies. So this is the observed and this is the expected. Like we were expecting, a researcher was expecting a frequency of 25, but he observed um, 20 for a particular position in a gene or a genome. You can say like this. Okay. Now the code is very simple. Chi2 underscore state and chi2 underscore p. This will print or this will calculate the chi2 statistic or the values, and this will print the p value, just like in case of the t-test we just observed little earlier. And the function will be states dot chi-square. Then if underscore observed, I will mention that this is the observed, and expected, this is the expected group, and we will run it. Now if you think the chi-square test statistic is 2, but the p-value is 0.3, what do you think? It is significant or not? No, it is not significant, because there is no exponential like in previous case exponent. So the real p-value is 0.36, which is far higher than the 0.05, uh, which is a threshold for the significance. So in this case, I would say that if you have this data, that means that the frequency I observed is just by chance, and it has no uh, significance, and I'm not going to, or you are not going to um, present this data with the statistical significance in your manuscript, because it failed to reject the null hypothesis. The equation is quite simple. It failed to reject the null hypothesis because the significance is not, or the significance is very low, uh, not in number. Actually, if the number is very low, then the significance will be higher. Okay, so here you can read it. The test does not rejects the null hypothesis. So uh, on this data, you can say that the chi-square test failed. Okay. Now, the third test we normally perform, that is called correlation analysis. In the correlation analysis, again, I'm simulating some gene expression data and protein abundance data. Um, I want to generate a data set that have some values for gene expression and some value for protein abundance. And I want to see, is there any correlation between the high expression and the high abundance of protein and the high expression of that protein or not. So, uh, First of all, I just write a code like gene expression data generate some random data, and then protein abundant also generates some random data. Okay. Now then we perform some correlation analysis and we call it Pearson correlation. So this is the function. We once again we will be interested to calculate two things: correlation and the, how to say, the p-score. So this is the function state.pearson, and for the gene expression and protein abundance, these are our two data types. The gene expr, this is the one data type, and this is another data type. Okay, and we will run it. Now, if you see here, the correlation coefficient is 0.79, but we are not interested in this coefficient, although remember the correlation everywhere in the correlation analysis, you have three values. If this is plus one, that means 100% positive correlation. If this is minus one, 100% negative correlation. If this is zero, no correlation. So if you see our value is 0.79, or in other words, if you round it, it's our value is almost zero point 0.8. So zero point 0.8 is near to plus 1. That means that our data shows positive correlation between the protein abundance and the gene expression. But whether this data or this result rejects the null hypothesis or not. So we will decide based on the p-value. So the p-value is 8.06, but look at here, it is 10 raised to power minus 12. Okay, so now when we calculate this, it will be a very lower value, or you can calculate it, the value will be extremely lower than this one, and you can say that there is a strong, or the extremely significant correlation. So this is the statistical significance of the correlation test. Now here, I'm going to visualize. If you want to visualize the correlation like the gene expression and the protein of abundance, then we discussed this kind of chart in my previous chapter 7. And let's see. Okay, so I'm not going to discuss it again. Okay, now if you want to perform these kind of tests, so remember you have to use for the t-test we use states.t-test underscore ind for independent. This is a function. For the chi-square test, we use state chi-square this function and this stats.pearson. If you don't know where I use, so let me show you here, state.pearson, and then name of our data set. The same we used this function in other two tests. Okay. Now, what actually is SciP state? SciP state is actually a package, and when you are working in Python or when you are working on Google Colab, you don't need to install the SciP state package individually. What we did in the start, if you come to the first command, we run it. So now you will understand the importance of the imports. So we import uh, state library from the SciP package. So already, if we failed or if we forgot to import this statement, then of course we will have some error, and our workspace may not be able to understand these kind of functions. Okay, that was for today, and I hope you understand like how to perform t-test how to perform chi-test and how to perform the correlation analysis. And I also tried my level best to provide you some theoretical background that that will help you to as a foundation if you are still not very clear on these kind of statistical test. You can perform your statistical test with different softwares like SPSS and many other, but you can also use this kind of Colab and use these. Even you can use my this notebook and calculate your own data. The difference will only be to upload your data, and that is very easy. As I told you here, just I am simulating a fake data, just like generating some data and then teach you on that. But you can just ask from Google how to upload your file to the Google Colab. They will provide you a statement. Just copy their statement, paste it here, and just run it. So you will be able to upload your data to this notebook. Then the next step you will need to do just to change these names. Okay, because maybe your data have some different labels. I the code reads untreated. Maybe in your case that is a different label. Okay. So thank you so much. If you are liking my videos, please thumbs up and share it with your friends, colleagues, and students. Thank you so much. Bye bye.